Hello. Today we are going to talk about muscle tissue. When we were in the tissue chapter, we mentioned that there are three different types of muscle tissue. So if we look, our first type, skeletal muscle, this is going to be the main focus of this chapter because most skeletal muscle is what causes you to move. It is striated. So it has those light and dark bands that we noticed from the tissue chapter. It's voluntary, meaning you can move it. You can move your arm around. You can move your hand, your fingers, your legs, your trunk. And it's controlled by neurons. So that nervous system, we're going to look at how skeletal muscle moves with these neurons. It's part of the somatic voluntary division. Somatic means muscle. Most skeletal muscle we move subconsciously. We don't always tell our muscles to move in a certain way. They're moving without you having to say left foot, right foot, left foot, right foot when you're trying to walk. Cardiac muscle. We looked at cardiac muscle in the muscle chapter, or the tissue chapter as well. Cardiac muscle is found in the heart. It is also striated, those light and dark bands, but it is involuntary. Cardiac muscle has its own pacemaker cells that initiate each contraction, meaning if the brain is not telling the heart to beat, the heart can still maintain its own rhythm. That's what that means. That autorhythmicity is that built-in rhythm. Hormones are going to change that rhythm, and so can neurotransmitters. These are chemicals that the brain uses or the nervous system uses to communicate. The other type of muscle is that smooth muscle. We'll also look into smooth muscle and kind of how it works as well today. Smooth muscle, it is non-striated. That is what makes it smooth. It is involuntary. We can't control how fast something is moving through our system. It is regulated by those autonomic neurons. It's also regulated by hormones. We find these in our blood vessels, airways, most organs, digestive, reproductive, urinary system. We're also going to find it in our skin that is attached to those hair follicles. That is what gives you that goosebumps when you're cold. So the function of muscles, they're going to produce body movements. They're going to stabilize body positions. When you're sitting here right now or sitting holding your head up, there's muscles contracting that is allowing you to hold your head up. There's muscles that are allowing you to sit up straight. So these are helping to stabilize your joints and help maintain those body positions. They store and move substances. So we have sphincters, external, internal, urethral and anal sphincters are good examples of those that allow or prevent something to pass through it. Your stomach, that temporary storage of food, that bladder, temporary storage of urine. So it's going to store things. It's going to help to move blood through those blood vessels. It's going to move food through that digestive tract, bile and enzymes through that whole digestive canal. And whenever a muscle contracts, it generates heat. That is thermogenesis. So it is helped to use to maintain your body temperature. That is why shivering, it contracts muscles and it helps to heat you up. So these are functions. Let's look at properties. We mentioned this in the tissue chapter, but muscles are electrically excitable. So they respond to electrical signals known as action potentials from the nervous system. They can respond to electric, like in the heart. They can react to chemicals like neurotransmitters, hormones. They're even going to react to changes in pH and the bodily fluids around them in the blood. They have this contractility, which is that ability to contract. They have this extensibility. Test question. Stretch without being damaged. Within limits, of course. Elasticity, another part of that test question. Returns to its original length and shape after a contraction of extension. So that extensibility allows them to stretch. That elasticity allows them to return to their normal shape. So let's look at what makes up a skeletal muscle. Skeletal muscles have hundreds to thousands of cells, muscle fibers, or myocytes. Myo means muscle. So whenever you see myo, we're talking about muscle. All muscle, skeletal muscle, has connective tissue that's going to surround those muscle fibers. They're going to have blood vessels because they need lots of nutrients. They need lots of oxygen. They need lots of glucose. And they need nerves because these nerves are going to tell these muscles when to contract and when to relax. So we're going to start. We've got that subcutaneous tissue of our skin. That is known as that hypodermis. That is made up of that areolar connective tissue and adipose tissue. This is also kind of considered that fascia that is found underneath that subcutaneous tissue, and it is irregular connective tissue, and it's going to line a lot of the body walls and limbs. Fascia supports and surrounds muscles. It holds muscles together, but it also allows for free movement of muscles, nerves, blood supply, lymphatic vessels, and it's going to fill spaces kind of between those muscles. So this connective tissue that wraps around muscles, we've got a lot of it. We're going to see some of these parts of words over and over again. So I'd mentioned it before, but epi is that outside. Epimysium, that's muscle. And then we've got that epi, that means that outer layer. So this wraps around that entire muscle. It's that dense, irregular connective tissue. Next is that perimysium. Peri is that kind of middle layer. What this perimysium, that's dense, irregular connective tissue, it's wrapping around tens to hundreds of muscle fibers, creating fascicles. I have a picture in a minute I'll show. And then endomysium. Endo is innermost. So it's that interior. So it's going to wrap around each individual muscle fiber. So let's look at this picture here. This first one. So what is going to wrap around, that was big, each in this entire muscle. This entire muscle here is wrapped in this epimysium. Next layer in, perimysium. This perimysium is creating a fascicle. So it is wrapping around a group of muscle fibers. So that perimysium creating that muscle fascicle. Now we get down to, it's not listed here. I'll write it. We have this endomysium. This is wrapping around, this is an individual muscle fiber. A muscle fiber is made up of myofibrils, which are lots of individual little parts. So let's look, here is a muscle fascicle. So we're coming down again, that muscle fascicle. So a muscle fascicle is a group of muscle fibers. That muscle fascicle has that perimysium. Now we have this muscle fiber. This muscle fiber is wrapped in that endomysium. And what makes up a muscle fiber is several myofibrils. These terms, the peri, epi, and endo, are going to keep coming. You'll see them again in the nervous system. So learning them here, we'll be able to apply and relate them later. Here we see that muscle fiber. You see all those myofibrils inside this cool electron micrograph. I love electron micrographs because they're so realistic, because they are real. Okay, fibromyalgia. If you don't know it, I'm sure you may have heard of it, at least heard the word. Fibromyalgia affects connective tissue that attaches to muscles, tendons, and ligaments. It's very painful. And these people get like tender points that you don't even have to push down where it's pain. They can have stiffness of tendons, muscles, and soft tissue. People who have fibromyalgia, they usually have severe fatigue because they're in so much pain that it's hard to sleep, which is that poor sleep. They get headaches. You don't get enough sleep, you get headaches from that alone. Depression, irritable bowel syndrome. And they sometimes get to the point where they have a really hard time just carrying out everyday activities. You can only fake feeling well for so long. These people usually need some significant stress reduction, get them exercising pretty regularly. They can apply heat to those areas, get a gentle massage. I've worked with patients with fibromyalgia before. It doesn't take much to cause quite a bit of pain. I've done acupuncture. It's another thing, your book doesn't talk about it, but acupuncture can work with fibromyalgia. You kind of stick needles into those tender points to try to help desensitize them. Because I wanted to come back up here and mention these connective tissues. All three of these layers, this epimysium, perimysium, endomysium, come together at the end of those muscles And they're going to form tendons that are attaching that muscle to the bone. They can also come together and form an aponeurosis. An aponeurosis is, again, connective tissue that can be worked to connect two different muscles together, like the epicranial aponeurosis. It's on the top of the head. It's going to connect the frontal and occipital parts of the occipital frontalis muscle. I wanted to talk about the ends of those before I forgot. So, muscles require a lot of oxygen and a lot of glucose and other ions, nutrients. So, we need blood supply. We need arteries. There's usually one or two veins that accompany every artery because there's a lot of waste products that are generated whenever a muscle contracts. We've got nerves that are called somatic motor neurons, aka muscle motor neurons. These neurons are coming from the brain or the spinal cord, and they're going to branch and attach to different muscle fibers. These arteries are going to break down into capillaries. Those are our smallest blood supply, blood vessels, because they're bringing those oxygen and those nutrients and they're removing heat and waste products from muscle metabolism. We'll go through some of these waste products today, and you'll go through them in a lot more detail in anatomy too when you talk about metabolism. So we're going to go down to the very microscopic level. So what is happening, what we see in each of those myofibrils that are inside of a muscle fiber? Excuse me. We have a sarcolemma. This sarcolemma is around this muscle fiber, and they create these T-tubules on this front area of this muscle fiber. So here we have this sarcolemma, which is this blue that is wrapped around this entire muscle fiber. Within this muscle fiber, we have these myofibrils. And then we get down to, I want to miss something, these T-tubules. These T-tubules, they're not labeled. I was looking for one that was labeled. There it is. Is this transverse tubule. T-tubules are these transverse tubules, and they back up to these terminal cisterns. So the T-tubule is this blue portion. These terminal cisterns are this kind of gold that I'm coloring over it in yellow. And all of these parts in between. Those are those terminal cisterns that kind of butt up to that transverse tubule or T-tubule. There we go. Within this sarcolemma, all of these individual myofibrils, around all of that, we have this sarcoplasm, which is the cytoplasm within a muscle cell. There's a lot of glycogen. Glycogen is a chain of glucose molecules. It's the way our skeletal muscles and our liver store glucose. They are the only two organs that can store glucose, skeletal muscle and the liver can. We use that glycogen. We can break that glycogen down into those individual glucose molecules to then make ATP, our energy. Myoglobin, something else in there. It is Myo means muscle, so it's only in skeletal muscle. It's only in muscle. And this is where oxygen, we actually store some oxygen in our skeletal muscles for when we need it. So when your muscles are at rest, relaxing, they collect some of that oxygen that is floating around in the blood and they store it and save it for when you go to use that muscle. So there's already there's oxygen readily available to be used. So here, skeletal muscle, I think I pointed it out in the tissues chapter, but if not, skeletal muscle has more than one nucleus. Why? Well, skeletal muscle starts as myoblasts. Several individual myoblast cells, and they're going to fuse together, but they all keep their nucleus. So as these myoblasts stay together, fuse together, this muscle fiber that is developing here is keeping all of its nuclei. So muscle cells have lots of nuclei because of the way that they fuse together during development. Muscular hypertrophy. Hyper means above normal. So our muscles are going to grow after birth. And they cause this enlargement of these existing muscle fibers. So muscles that get larger and larger, muscle hypertrophy is because of that increase in myofibrils, mitochondria, sarcoplasmic reticulum, and the other organelles that our muscle cells need. There's more myofibrils inside that muscle cell. It's going to happen during childhood because of growth hormones and other hormones that are going to stimulate the skeletal muscle growth. Testosterone in males are going to produce even further muscle enlargement, which is why males can develop larger muscles than females, is because of that testosterone. We keep a small amount of satellite cells. Satellite cells are stem cells for muscle. So we keep a small amount of those stored in mature muscle cells. They can kind of fuse to muscle cells we already have, and they can help to regenerate functional muscle fibers. But if a muscle is damaged, the problem is because we only have a few of these myoblasts or these satellite cells, sometimes we don't have enough. There's not enough satellite cells, and it can actually cause muscle damage or degeneration. Those tissues are going to undergo fibrosis, where there's scar tissue formation where that muscle was damaged. Like mini micro tears that are created in muscles from exercise. We'll go into this a little later in this section, but we have enough satellite cells available to fix these little micro tears. If you do have like a full on tear to a muscle, we don't have enough of those satellite cells available to fix it. So scar tissue is going to form instead. Atrophy. Atrophy is a decrease in size of muscle fibers. it causes this loss of myofibrils. There's two different types. There's disuse atrophy. Disuse is because you're just not using. You don't use your muscles, they get smaller. You lose your strength. There's also denervation atrophy. So the nerve that goes to that muscle cell doesn't work. So if that nerve that doesn't go to that muscle cell doesn't work, could be disrupted, could be cut. There's a lot of things that could happen that could cause it. You're no longer able to use that muscle cell. So those muscle cells are going to shrink. They're going to get smaller because there's no nerves telling it to contract. So those myofibrils, as I mentioned, we have tens, hundreds, thousands of them inside of our muscle cells. These are what actually contract in skeletal muscle. These myofibrils give muscles that striated appearance. We have a sarcoplasmic reticulum. This is where we calcium when our muscles are at rest, is that sarcoplasmic reticulum. Those terminal cisterns that I pointed out, those are part of that sarcoplasmic reticulum. So I want to relax. Here, I have it here. Stores calcium ions, that sarcoplasmic reticulum, when muscles are relaxed. They are released during muscle contractions, and we will see how that works. So here's the setup. We have these sarcoplasmic reticulums right here, kind of outlined in blue. This and all of those little purple dots, those are our calcium ions that we have stored within this muscle cell. We have each of these individual myofibrils here. This is the contractile unit of that muscle cell. So now we're going to go into these myofibrils here. So our myofibrils are made up of these filaments. These filaments are proteins. We have both thin filaments and thick filaments. These thin filaments are actin. These thick filaments are myosin. These work together to contract a muscle. We have two thin filaments for every thick filament. Within these filaments, they are set up into sarcomeres. Sarcomere is the part of that myofibril that is going to contract. Okay, I wanted my picture because it's easier to see. A sarcomere goes from a Z disc to a Z disc. So these blue kind of zigzag line on each side, those are, this is what makes up that sarcomere right here. Then we have, trying to see where else I want to go with this, sorry. We have our M line. I'm trying to draw in purple here, not very well. This M line goes down the middle. M for middle. We have an A band that is found on either, or that follows the entire length. I know what I want to do with this. The entire length of these thick filaments. This darker red line here these darker red lines, those are my thick filaments, and that A band will go from thick filament to the other end of my thick filaments. My H band, pick a color, any color. My A band, or no, A band, not A band, H band. H. Haha, H. That H band is where we have just thick filaments. So those thick filaments and nothing else. So that is my H band. My I band, that's what I was looking for before. My I band is just thin filaments. Nothing else here, just these little bits of thin filaments from one side to the other. That is my I band. So if we go down from this drawing to this actual details of what we have here, we have our thin filament, which is in yellow. This thin filament in yellow is my actin. We have our thick filaments in red. That is my myosin. We have the M line, color, M line in the middle. That just designates my middle. My sarcomere goes from that Z disc on one side to my Z disc on the other. We have what's holding these M or these myosin in place. We have this protein here that is called titin. Looks like a spring. That titin is helping to hold this muscle, these myosin in place. So when you go to stretch a muscle, this titin is helping to prevent that overstretching to tearing in a muscle. So we have that H band that is just thick filaments, that A band, which goes from the end of one thick filament to the other end of thick filaments. And then we have that I band, which is just thin filaments. And then we have this zone of overlap where we have both thick and thin filaments. So here is an electron micrograph of a sarcomere. You can see where the thick filaments are, the overlap, this zone of overlap here. Didn't really work with green, but whatever. And then you can see where you have just thick filaments in the middle. You can see that M line very well right through here. And then you also see those Z discs on either end very well. So these myofibrils are made up of those actin, that myosin, that titin. We've got those Z discs on either end. Those are other proteins that we're not going to go into as much detail about. But what actually contracts in that muscle is that actin and that myosin working together. So I mentioned that myosin is that thick filament, and that actin is that thin filament. So if we look at myosin, myosin is kind of described as two golf clubs twisted together. We have a tail on that myosin that points towards that middle or M line. So let's look at this. Here are these myosin heads. There's usually two golf clubs tied together on that tail, this tail here. That is how we describe myosin. On the myosin, let's look at this quick. We have this actin binding site. So this is where myosin will attach to that actin. We also have an ATP binding site. Muscles need energy to contract. So we have to have a place where ATP can bind to allow this contraction to happen. And we can see all of these little golf club heads throughout here. There's hundreds of them that are helping to form this myosin protein. And here's our M line in the middle. So I mentioned all of that. We've got tropomyosin and troponin. These are two other proteins. These are two proteins that you do need to know. So there we go. Tropomyosin and troponin, they're considered regulatory proteins, and they are found on that thin filament, that actin. So in a relaxed muscle, the tropomyosin is going to block these myosin binding sites here. We have our troponin that is attached to this tropomyosin. Calcium binds to that troponin. When calcium comes in, so we have a calcium comes in. What it's going to do when this calcium binds to this troponin is it's going to move this tropomyosin out of the way so myosin can bind to that binding site. As soon as calcium goes away, cross it out, troponin is going to move that tropomyosin back over that binding site so myosin can no longer attach. I've got some videos that I'm going to show you, or I will attach them, we'll see, to kind of help to show this whole process. So I mentioned that Titan, it's kind of like a spring, and it goes from the Z disc to that M line, and it's helping to kind of hold that myosin in place. It's very elastic. So it can be stretched at least 4 times its resting length and spring back unharmed. So this kind of helps prevent overstretching of muscles. And it helps, we think it also helps to return muscles back to that resting length. So these are some of the other proteins. I'm going to mention them. We have alpha actinin. We have myomesin that makes that M line. Nebulin, dystrophin. So these are just other proteins that are helping to kind of make up that entire sarcomere. So this is going through all the different proteins that are making up the contractile portion, the regulatory, and the structural aspect of the entire sarcomere. This goes through the parts of the muscle fibers, those epimysium, perimysium, endomysium, down to the sarcomere and the filaments, that actin and the myosin that help to make up that entire sarcomere. These are nice tables to just kind of show where everything is at. Now we get into our first physiology section of anatomy. So we use the sliding filament mechanism or sliding filament theory of how muscles contract. Muscle contraction happens because these myosin heads are going to attach and walk along that actin filament. They're going to pull that thin filament, that actin, towards that M line. So that thin filament's going to slide inward. The I band in the H zone narrows. It gets smaller and eventually can disappear depending on how much that muscle is contracted. So that the length of the thin and the thin filaments do not change. It's just that they're moving directions here. So these thin filaments, they're attached to that Z disc. So those Z discs are going to come closer together when that sarcomere shortens. So we have two sarcomeres. We have a Z disc, a Z disc, a Z disc. This is a relaxed muscle. So when our muscles start to contract, these myosin heads are attaching to this actin here. And they're going to start to walk along this actin towards that Z disc on both sides. They're going to keep walking. If we'd have a maximally contracted muscle, such as in the bottom, you can see how these myosin heads made it all the way to these Z discs. That is them walking along that actin filament to shorten that muscle. So if we look at the microscopic picture here, you can see this M line. The M line never changes, stays in that middle every time. But what is changing? This H band here got smaller. That H band disappeared. We have even the A band is pretty hard to see. The A band, that's all we have is this A band really. Take that back. The A band. We've lost that H band, and that I band has gotten smaller as well with just thin filaments. So let's go through step by step how this happens. Paige puts it in words. I'm going to go through pictures. So those myosin heads hydrolyze ATP. So what they're going to do is they take this ATP, they're going to use this ATPase enzyme. Thought it said it up there, sorry. This ATPase enzyme, and they are going to break ATP into ADP plus phosphate. By breaking that ATP plus phosphate, one of their byproducts is energy. So now we've created energy by breaking up ATP. This energy gets stored in this myosin head. Next step, this myosin head is going to bind to that actin. So it forms this cross bridge. Cross bridge meaning this myosin and this actin here. This myosin head came in contact with this actin. That forms the cross bridge. Step 3. This myosin head pivots because of the energy that was created by breaking that ATP into ADP plus phosphate. We lose the phosphate when we attach the myosin head. it allows that myosin head to pivot its head with that energy created that was stored in that head from breaking down that ATP. That is called the power stroke. Now, for this myosin head to break off of this actin, so to release this myosin head here, what we have to come in and what we need is to release that ADP. That ADP comes off. And an ATP has to bind. So we have to bind a new ATP for that myosin head to release. So that cross bridge detachment. Without ATP, that myosin head cannot release. That is the main steps of this cross bridge. So let's look at this happening here. So for a muscle to contract, we're going to look at this cross bridge cycle. So we have our sarcomeres. Our sarcomeres will contract. We break it all the way down. Now we're looking at this actin and these myosin filaments, that thick and that thin filament. You find the thick filaments in red, the thin filaments in yellow. So let's look at what happens. So we've got these little calcium ions that are coming in and attaching to that troponin. When calcium binds to troponin, it is going to move these tropomyosin out of the way. Now we have our actin binding sites, or the myosin binding sites. So this myosin head that is bound to actin, we have a new ATP molecule that comes in, causes that myosin to release. It's going to break that ATP into ADP plus inorganic phosphate. That puts energy into that myosin head. So for our cross bridge cycle, step 1, form that cross bridge. Myosin is going to bind to actin. We release that phosphate when we bind. Step 2, that power stroke. So the energy that we created from breaking up ATP allows that myosin head to do a power stroke. Step 3, cross bridge detachment. We need that new ATP molecule to come in, bind to the myosin head to allow that detachment to happen. Then we have to reactivate that myosin head. So we break down that ATP to ADP plus phosphate, which re-energizes that myosin head and puts it back into its starting position. So for our muscle to contract, we can see how these myosin heads are walking along this actin. We can see it from a whole sarcomere point of view. So now myosin heads are gonna start walking along this actin, causing this muscle to shorten. When a muscle is relaxed, that calcium gets released from that troponin, which moves that tropomyosin back over that myosin binding site. That myosin binding site stays covered, that muscle stays relaxed Until the next time. And that was a nice drawing or nice video of this entire process happening. I will also post this into Blackboard so you can see this, go back and watch this, just this portion of the video as often as you would like. So that went through everything I went through. So within the sliding filament theory, we've got those elastic portions. We have that tighten. It's kind of like a spring. So that nice little spring that went along. There's connective tissue that allows muscle to stretch. There's tendons that attach muscle to bone. These are all helping to hold this muscle in place. It's also allowing that stretching to happen. So as the skeletal muscle starts to shorten, it's actually going to pull on that connective tissue covering. It also pulls on those muscles and those tendons. Those tendons become tighter, taut. And that tension that's kind of put through all of that is actually what causes that body part to move, that pulling on that tendon pulling on that bone, and then that's causing that bone to move. So excitation, contraction, coupling. So we kind of looked at it in that video that I just showed you, talked you through. If you watch it on yourself, it has its own recording that for some reason was not playing, but that's okay. So if the more calcium, that's increase in calcium that's being released from that sarcoplasmic reticulum starts that muscle contraction. As that calcium gets put back into that sarcoplasmic reticulum, it can decrease that, it can stop that muscle contraction. So that calcium is stored in that sarcoplasm, in that sarcoplasmic reticulum. So calcium in the sarcoplasm is very low. We keep it in that sarcoplasmic reticulum until we need it. So a muscle action potential. It's going to, a nerve is going to tell a muscle that it needs to contract. We'll go through the steps of that here shortly. That's the next part of the physiology. When the nerve tells a muscle to contract, it causes calcium to be released from the sarcoplasmic reticulum. That calcium is going to bind to that troponin, which moves that tropomyosin out of the way and allows that myosin to bind to the actin. So that triad, that T-tubule with those terminal cisterns of that sarcoplasmic reticulum. That T-tubule and those terminal cisterns are usually kind of linked together. We have voltage-gated calcium channels. Voltage-gated, meaning they respond to a change in charge within that cell. So if that cell that usually sits pretty negative becomes more positive, it causes those calcium channels to release calcium. We get calcium back into that sarcoplasmic reticulum through calcium ATPase pumps, meaning we use ATP to put calcium back into that sarcoplasmic reticulum. We store that calcium in that sarcoplasmic reticulum when our muscles are relaxed. So, what we just went through. Calcium is stored in the sarcoplasmic reticulum. They respond to a change in charge within our cell. So we have these voltage-gated calcium channels. When our cell changes its charge, usually more positive, it's going to release these calcium ions out of the sarcoplasmic reticulum. Those calcium ions are going to go down and bind to this troponin down here. When the calcium binds to the troponin, it causes that tropomyosin to move off of those myosin binding sites. Once that happens, it allows myosin to bind to this actin. And that is when this cross-bridge cycle can happen that allows a muscle to contract. So, an action potential comes across, changing that charge. When that charge changes, this calcium gets released. The calcium that gets released will bind to that troponin, which moves that tropomyosin out of the way and allows these myosin heads to bind. And it'll allow for this whole process to start. So let's look at what is happening here.
Structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as the sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As the pencil travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
It's another video I'll post for you, another link that you can look at in more detail. So we have ways that we can actually monitor muscle contractions and these nerves that are sending this information down to muscles. It's called electrodiagnostic, highlighter, electrodiagnostic medicine. We use it to diagnose neuromuscular disorders, so between the nervous system and the muscle system. So they can do what is called a nerve conduction test. What they're doing is they're measuring nerve impulses, those action potentials that come from the brain and the spinal cord and go out to the muscles. They can use it to diagnose carpal tunnel syndrome, herniated discs, sciatica. They use an electromyography. Myo means muscle. So they take that thin they take a thin needle and a recording device. They place it through the skin into that muscle specifically. And then they can monitor what is happening. If the muscle is resting, there's no electrical activity. If there's electrical activity present, it can cause a muscle contraction. So they can diagnose those muscle dystrophies, myasthenia gravis, or amyotrophic lateral sclerosis. We'll go through what these are at the end of this chapter. So here's kind of an example of what they're doing. They're taking this needle, sticking it down into a muscle, and they're recording that muscle. Is it receiving any information from the brain? When a muscle contracts, they can detect that signal that was sent from the brain through those nerves down to that muscle. Here's another interesting one. Rigor mortis. You know how I kept mentioning if we need ATP for that myosin head to detach from that actin, that actual detachment process of the cross bridge to happen. After we die, our cells can no longer make ATP. Our cells also become pretty leaky. So what was once holding that calcium into the sarcoplasmic reticulum just kind of relaxes and calcium starts to flow out. So calcium ions leak out of that sarcoplasmic reticulum. They bind to the troponin, which causes the tropomyosin to move out of the way. And if the tropomyosin moves out of the way, myosin heads are going to bind. It's just an automatic process. Those myosin heads are going to bind. So if those myosin heads bind, they can start the contraction process. They do their power stroke, move their head, but there's no ATP. So those myosin heads are stuck on that actin. And that is what causes those muscles to go in that state of rigidity, where that body stiffens up, everything gets stiff and rigid. It starts usually 3 to 4 hours after death, and it lasts about 24 hours. It can last longer than that, but that's just kind of they can use that to kind of determine a time of death even. Eventually, those muscles relax because of proteolytic enzymes from those lysosomes. Again, our cells getting leaky start to leak out, and it starts to break down that actin and that myosin, which causes those cross bridges to break. This is what causes rigor mortis and what causes the end of rigor mortis. So let's look at this length tension relationship. So how much of a force we can generate in a muscle contraction is all going to depend on the length of the sarcomere at the beginning of that contraction. That zone of overlap that I pointed out in that sarcomere, where that zone of overlap is to generate that most forceful contraction. That zone of overlap between that thick and thin filament, it extends from that H zone to one end of a thick filament. So as a sarcomere of a fiber, they get stretched, that zone of overlap is going to shorten. A skeletal muscle, when it is stretched to 170% of its optimal length, that is when there is no overlap between that thick and thin filament at all. Meaning the fiber cannot contract because there's no place for that myosin to bind to that actin because of how stretched that muscle is. As that sarcomere gets shorter and shorter than its optimal length, that tension also decreases because we can't generate as strong of a contraction when that muscle is already somewhat shortened. So if we look at this nice picture here, our optimal length is between 100 to 120, usually between 80 to 120% of its optimal length or of overlap is where we can generate the strongest contraction. That overstretch, this is that 170 we mentioned on that slide before. This is where you can kind of see those thick and thin filaments aren't in contact with each other. There's no place for them to come in contact with each other, I should say. So at this point, we can't generate a contraction at all. At 60% of our optimal length is where we can no longer generate a contraction either, because we're getting to the point where those myosin heads are butting up against those Z discs. So there's no place for that muscle to go. Let's get into the second part of this entire physiology process of a muscle. We're going to look at this neuromuscular junction. So we have that somatic motor neuron. This is the neuron that comes from the brain and the spinal cord that is coming out to these muscle cells that tells these muscles to contract. We have the neuromuscular junction. This is the space between the nerve and the muscle cell. The synapse is where this communication between two neurons or between a neuron and its target cell. So in this case, it's going to be between a neuron and a muscle cell. The synaptic cleft is that gap. The neurotransmitter is a chemical messenger. For this case, we write ACH, acetylcholine is our neurotransmitter for skeletal muscle. And then we have our axon terminal, which is that end of the motor neuron that's going to come down to that skeletal muscle. We'll finish going through all of these different parts and then I'll show it on a picture. We have our synaptic end bulb. That is the end portion of that neuromuscular junction. Synaptic vesicles. We've mentioned vesicles. Vesicles are those membranes and closed sacs that are going to hold that neurotransmitter, that acetylcholine. That acetylcholine is held in those synaptic vesicles that then get released into that neuromuscular junction. The motor end plate is the muscle portion of this. On that motor end plate, we have acetylcholine receptors. These are ligand, no, I want a pen, not pencil, ligand gated channels. A ligand-gated channel is a channel where we need something to bind to that channel to cause it to open. Well, these acetylcholine receptors sit on these ligand-gated channels. So when that acetylcholine binds to that acetylcholine receptor, it's going to open that channel. And then we have junctional folds. These are just kind of these deep grooves. That picture we saw before that kind of go like this, it works to kind of increase the surface area. That's what it does. So let's go to the picture first. This whole thing was what happens. I'm going to explain it out and then I'll show a video. So this is the neuromuscular junction. We have the end of the nerve, which is the synaptic end bulb. And then we have the motor end plate, which is the muscle that is involved. That is that neuromuscular junction. We have these synaptic vesicles that are kind of shown in green that have red inside. That is that acetylcholine. The sarcolemma, this is where this muscle is attaching to. Let's try to see. Okay. So let's look at what happens. And I would like to draw a picture. So I have my nerve coming down. I have my muscle coming across. Along my muscle, I have these ligand-gated channels. Whoops. These little channels. They are currently closed. On my nerve, I've got these other channels here coming in on both sides. I have synaptic vesicles found within my nerve. Inside those synaptic vesicles, I have acetylcholine. So, a nerve is sending down this nerve impulse that is saying to this muscle, telling this nerve to tell this muscle to contract. So, when this action potential comes down, this nerve impulse, it causes these channels to open. When these channels open, calcium is going to come inside this synaptic end bulb of this nerve. This calcium here is going to cause the synaptic vesicles to bind, to the end of this nerve and release acetylcholine out into this synaptic cleft, this opening between this nerve and this muscle. This acetylcholine is going to bind to these ligand-gated channels. When this acetylcholine binds, it is going to open these channels. So this channel here is just going to open. I'll draw just a line down. That one opens and that one opens. So now that these channels have opened, what is going to happen is, looking for a color, red. Sodium is going to come rushing into my cell. Sodium starts coming in. It's going to cause, got other channels that can open on my cell here. These channels are going to open and they're going to cause potassium to leave my cell. When potassium leaves the cell, these acetylcholine here, whoops, I'm deleting the whole thing. That's okay. Is going to release off, which is going to cause this channel here to close back up. So I'm going to draw a graph up here, down here. I've got a graph. My muscle cell tends to sit at about negative 70 millivolts. So, when this process is happening, this calcium comes in, causes this acetylcholine to be released out from the synaptic vesicles to the synaptic cleft, which causes the acetylcholine to bind to those acetylcholine receptors, which causes the sodium channels to open. During this process, my cell, when sodium starts coming in, my cell becomes more positive. It can get up to about positive 30 millivolts. This is when those potassium channels open. So now my cell, potassium is leaving, potassium's positive, so it's causing that cell to become more negative again. And then we move, we've got other channels that are they're pumps. They're going to move sodium back out and push potassium back into that cell. That is what is happening in this area over here that goes down a little more negative. These potassium channels, it takes a little longer for them to close, which is part of the reason why that cell becomes more negative than that negative 70 millivolts. But we also need to move that sodium back out and that potassium back in. So that's going to affect this whole area. So I've got a little video down here. Turn the volume on my computer on again. Let's
travel from the brain or spinal cord, trigger the contraction of skeletal muscles. Step 2. Voltage terminals open. The calcium diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, the exit potential propagates along the sarcolemma. Transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholine esterase to acetic acid. Choline is then transported into the axon terminal for the synthesis of acetylcholine.
Oops. There's different models of how this works. Some show that the sodium and the potassium channels are the same. Other models show that there's different channels for sodium and potassium, but it's still the same concept where sodium enters into the cell, potassium leaves the cell. These are what cause this entire process to occur that allows this action potential to happen. So this is a really cool electron micrograph that kind of shows how these nerves branch so they can communicate with every single muscle fiber. So let's go through the entire process. An action potential comes down. Calcium comes into that nerve and that nerve terminal that causes that acetylcholine to be released. Acetylcholine binds to the acetylcholine receptors, which opens these channels, which allows sodium to come in. When sodium comes in, our cell becomes more positive. That change in charge causes these voltage-gated calcium channels to open and calcium to be released from the sarcoplasmic reticulum. When these calcium ions release from that sarcoplasmic reticulum, they're going to go bind to that troponin. When they bind to that troponin, it moves the tropomyosin out of the way. It allows those myosin heads to bind to that actin and allows those cross bridge formation and that power stroke to happen for that muscle to shorten. As that cell becomes more negative, as I drew on that graph picture before, that's what causes that calcium to be released from that sarcoplasmic or from that troponin. That troponin moves the tropomyosin out of the way. The calcium is then pumped back into that sarcoplasmic reticulum and that muscle relaxes. So that is the entire process from the beginning of a nerve action potential all the way to that muscle relaxation at the end. Here's some cool stuff. Plant toxins. Botulinum toxin. Clostridium botulinum is the bacteria that causes. It prevents those synaptic vesicles from being released. We don't release those synaptic vesicles, acetylcholine is not going to be released, which means there's no muscle contractions that are going to happen. This toxin can be found in canned foods that may not be sealed correctly. And this toxin can be lethal. A very tiny amount is needed to cause death by paralyzing skeletal muscles, including that diaphragm. That diaphragm stops working, you can no longer breathe. Here's where it gets interesting. Botulinum toxin, that's Botox. People use Botox for cosmetic treatments on facial wrinkles. They're paralyzing their skeletal muscles with the use of Botox to get rid of the site of wrinkles. But it does have some medical uses. People who have cross-eyed or what is known as strabismus, blepharospasm, uncontrolled blinking, or even spasm of the vocal cords. You could put some Botox in there, paralyze those muscles, and people no longer have that cross-eyed, that uncontrollable blinking. People can use it for chronic back pain from muscle spasms. but it can be a lethal toxin if not used correctly. But people tend to use it for cosmetic reasons. Another one, curare. This is another plant derivative. It is South American Indians used to use curare on their blow darts to paralyze the muscles of animals to help them catch their prey because it blocks the binding at the acetylcholine receptors. So those ion channels that allow sodium into that muscle cell cannot happen. Interesting part, curare. It is now used in surgeries to relax muscles. When they put you to sleep under the general anesthesia for a surgery, they are also paralyzing your muscles because muscles can sometimes do random twitches. And if they're working on something, they don't want a muscle to randomly twitch and cause more damage in the long run. So they paralyze your muscles when you're in surgery. They use anticholinesterase agents to help reverse the process. They're the anticholinesterase agents. This actually slows the removal of acetylcholine from that synaptic cleft. So it allows that action of the acetylcholine in that muscle contraction to have a longer effect. People who have weak muscle contractions, that can help strengthen them. Can be used to treat myasthenia gravis. It's also used for that antidote to that curare poisoning. So after surgeries, that's where I was originally going, but after surgeries, they need to reverse that paralyzing of your muscles. So they use this anticholinesterase agents to reverse the effects of the paralyzing of your muscles during surgery. So our muscle metabolism. How do our muscles make energy? We have three different ways our muscles can make energy because we use a lot of ATP. ATP is used for muscle contractions. It is used to pump that calcium back into that sarcoplasmic reticulum. And we also store a little bit of ATP that can give us a few seconds of a muscle contraction. So we have to make enough ATP to maintain muscle contractions. So we use creatine phosphate. This is something that is unique to skeletal muscle or unique to muscles. We have anaerobic glycolysis and aerobic respiration. So creatine phosphate. I want a picture. Creatine phosphate. We make ATP. What we're going to do is take this ATP, we remove a phosphate. So we take this and we do ADP plus phosphate. We take this phosphate and we attach it to creatine, and then we keep this ADP. We keep this creatine phosphate in our muscles in that sarcoplasm, and we leave it there until we need it. When we go to do something, this usually gives us the first 15 seconds of our muscle contraction. Because what we do when we need that ATP, we take that phosphate off of creatine and we put it back on that ADP to make our ATP. So that creatine phosphate is one of our ways to make energy. We take that ATP, we hydrolyze it, we break it down to ADP plus phosphate. We take that phosphate and attach it to creatine. The enzyme creatine kinase is the enzyme that helps make this happen. So again, it gives us about 15 seconds worth of energy. Creatine supplementation. We do get creatine from milk, red meats, and some fish. We need to take in about 2 grams of creatine to make up for what we lose through the urine. There have been some studies that show that creatine supplementation can help with athletic performance. The problem is by taking in creatine, it decreases your body's making of creatine. And we don't know if there's any long-term effects to it. It causes dehydration, and it can possibly cause kidney dysfunction. Next step, anaerobic glycolysis. We mentioned it a little bit in the beginning chapters. But what we do, anaerobic means without oxygen. So this is when we're not using oxygen. We put glucose molecule into glycolysis. Glycolysis is going to break down glucose into 2 pyruvic acid molecules. This all happens in the cytosol of a muscle cell, of any cell. And we produce 2 ATP molecules from 1 glucose molecule. No oxygen, as I mentioned, anaerobic, and means without oxygen. We use this when there's not enough oxygen in that muscle cell. The problem is this pyruvic acid can be converted to lactic acid. That lactic acid is will diffuse out of those skeletal muscles, get into the bloodstream, where it can go to the liver and be converted back to pyruvic acid or glucose, or our cardiac muscle can use this lactic acid to make energy. Cardiac muscle is the only place in the body that can use lactic acid to make energy. When it's produced very quickly, it can accumulate in those muscles. And this is what's thought to cause muscle soreness. Again, we use this when oxygen levels are low, but it can only give us about 2 minutes of energy. That's it. So that creatine phosphate gives us 15 seconds, anaerobic glycolysis gives us about 2 minutes. Aerobic respiration. So, aerobic respiration starts with glycolysis. So, we're going to take those pyruvic acids that were created from glycolysis, and they're going to go into the mitochondria where they're going to go through aerobic respiration, which is the Krebs cycle and the electron transport chain. You'll go through these in the metabolism chapter, and anatomy 2 is just mentioned here. Through this, we make ATP, carbon dioxide, water, and heat. This is slower, but it gives us 30 to 32 ATP per glucose molecule. Again, we can get oxygen from the blood, but we also store some oxygen in that myoglobin. Myoglobin and hemoglobin are oxygen-binding proteins. You'll learn more about the hemoglobin in anatomy 2. We get enough ATP through aerobic respiration. It can last several minutes to hours or more. We can provide pretty much all the ATP our muscles need through aerobic respiration. Muscle fatigue. Muscle fatigue is that inability of a muscle to maintain a forceful contraction after a prolonged activity. If you've ever gotten to the point where you can no longer contract a muscle, you have actually worked your muscles to the point of fatigue. You know what I'm talking about. If you've never done it before, it's kind of interesting to try to find that point where your muscles just can't do it anymore. There's two different kinds. We have the central fatigue, and it's caused by changes in that central nervous system. So the brain or the spinal cord. We don't know what causes it, but we think it can be a protective mechanism that's working to protect from muscle damage. So stop that person before that muscle damage occurs. We don't know for sure what causes muscle fatigue. Maybe not enough calcium, depleted that creatine phosphate, ATP levels in fatigued muscles. They're not much lower than those in resting muscles, which is kind of interesting. Maybe not enough oxygen. You've depleted all your glycogen stores. All your nutrients are gone. Too much lactic acid and ADP. Failure of that action potential. Failure of that acetylcholine release. There's a lot of different thoughts behind what causes muscle fatigue, but we're not really sure. Oxygen consumption after exercise. So after you're done exercising, if you've ever run across a parking lot or something, and after you finish, you still have that increased breathing rate after you have finished running. What we're trying to do is we're trying to restore oxygen levels, nutrient levels back to what they were before we started exercising. It can take anywhere from a few minutes to a few hours. It's also called oxygen debt. So we're trying to restore oxygen levels to our muscles, restore that myoglobin where we store that oxygen. We're trying to convert that lactic acid back to glycogen in the liver. We need to remake creatine phosphate, and we need to replace that oxygen from that myoglobin. This is all happening after we finish exercising. So after you finish exercising, there's usually very little glycogen that is technically made from lactic acid. Most of it comes from carbohydrates. Most of that lactic acid gets converted back to pyruvic acid and used for ATP production through the aerobic respiration. We have that elevated body temperature. Elevation in body temperature is going to increase the rate of chemical reactions. So we're increasing the rate of ATP production. Tissue repair can also happen. Also, another name for it is recovery oxygen uptake, because we're recovering and trying to get all of our stores for all of our muscles back to their resting levels. So muscle tension, all this other cool stuff. So action potentials are always the same. They give that the action potentials are the same every time it tells that muscle to contract. But what if we want a stronger contraction? If we want a stronger contraction, we need to increase the frequency or the number of impulses sent from that nerve to that muscle cell. This is the way to get a maximum tension within that muscle. The total tension is going to depend on the number of muscle fibers that are contracting in unison. So let's look at some of this. Motor units. All of our skeletal muscles are broken up into a motor unit. A motor unit is all of the muscle fibers that one neuron is going to make contact with. So when that one motor unit, that one neuron that goes out to all of these different muscle fibers, stimulate that muscle fiber, stimulate that, or a stimulation from that one neuron is telling all of those fibers that it's attached to contract. And it's kind of dispersed throughout that muscle rather than being clustered together. And there's a reason behind it because it's trying to prevent that muscle fatigue. So only certain parts of or certain fibers and muscles contract when we tell a muscle to contract. We do have some whole muscles that control precise movement. So they have very small motor units. Speaking, that voice box. The muscles that control your eyes, that allow you to move your eyes around. We have some skeletal muscles that have a motor unit of 2,000 to 3,000 fibers. Your back, it just kind of depends on where they're located. So this is the picture I was looking for earlier. We have these two neurons that are coming out, and you can see in the green, you can see which muscle fibers that green is attached to. And then the purple, you can see which fibers the purple is attached to. The two muscle cells this purple is attached to is one motor unit. The three muscle fibers this green is attached to is a different motor unit. So a twitch contraction. We're going to put all of these together here shortly. A twitch contraction is a brief contraction of all muscle fibers in a motor unit that respond to a single action potential. So one motor unit gets stimulated, so every fiber that is attached to that one motor unit, that one neuron, is going to contract. It's usually done in a lab. We use a myogram to record it. We're going to have that latent period, so that brief delay between that stimulation and that muscle contraction, about 2 milliseconds. And it has to do with that action potential, those calcium ions being released from that sarcoplasmic reticulum, even that space between that nerve and that muscle cell, that acetylcholine has to diffuse across. We also have this contraction period. That is when that cross bridge formation happens and that muscle is actually contracting. The next step is the relaxation phase. So calcium is being put back into that sarcoplasmic reticulum. Those myosin heads are detaching from that actin. That troponin is moving that tropomyosin back over. And then we have what's called a refractory period. This refractory period is the time where a skeletal or where a muscle is not just skeletal muscle, where a muscle cannot respond to another stimulus. So if two stimuli are received, one right after another, the muscle will respond to the first one, but not the second one. The refractory period is different in different muscle types. Skeletal muscle is about 1 millisecond. Cardiac muscle, it has a very long refractory period of about 250 milliseconds. So it's all going to depend on what is happening. So we have that latent period, that contraction, that relaxation, and then that refractory period before the next one comes in. So that frequency of stimulation, that second stimulus occurs after that refractory period, but before that skeletal muscle has fully relaxed, that'll actually generate an even stronger contraction than the first. This is where these wave summations will kind of come into play. And it is the frequency at which these action potentials are coming to this muscle cell. Picture. So a single twitch gives us, we have this one action potential that gives us this one contraction. We have two action potentials that come in. We get our first muscle contraction. Our muscle hasn't fully relaxed, but it is far enough through that action potential process to where we can generate another contraction, which means we get a stronger contraction out of it. Unfused tetanus means we get one contraction, starts to relax, generate a stronger, generate a stronger, generate a stronger. Four action potentials just sequenced in the right way will generate a very strong contraction. Fused tetanus, on the other hand, action potentials are coming in so rapidly that the muscle generates this maximal contraction and holds that maximum contraction until the action potentials stop. So our motor units, we tend to use a recruitment to help generate stronger contractions. So this motor unit recruitment just means the number of active motor units is going to increase as we get more action potential sent. So different motor units get stimulated at different times. So say you're doing a bicep curl where you're flexing your arm, your hand up towards your shoulder. Each time you do a bicep curl, a different motor unit is working. And we usually start with the smallest, weakest motor units first, and then we'll get to the stronger and stronger ones as we need a stronger force to do something. Why are we doing this? Because we're trying to prevent muscle fatigue, and it allows contractions to be held for long periods of time. One contracts while the other relaxes. The smaller the motor unit, the more fine, precise movements we get. The larger the motor unit, the less precision we get. So, jogging. I love how your book loves to talk about aerobic dancing. Different types of aerobic activities, your muscles will go through that aerobic respiration. Weightlifting is anaerobic through that glycolysis. So aerobic training stimulates muscles, proteins to increase in size. Or anaerobic does, sorry. Anaerobic causes an increase in muscle size. So people who do anaerobic training, weightlifting, they're working to increase their muscle mass. Aerobic training works to build endurance, so you can do activities for longer periods of time. There is different training methods out there that uses a combination of aerobic and anaerobic. So they're using both types of training as a way to build strength and endurance at the same time. Muscle tone. This is another interesting one. Your muscles are sitting in partial involuntary contractions. So all of your muscles have some sort of definition to them, some sort of shape to them, because they are holding a partial contraction. And this is because between the neurons in the brain and the spinal cord that excite those muscles. So when motor neurons that go to these skeletal muscles get cut, they become flaccid, flat. There is no muscle tone to them whatsoever. So to maintain this muscle tone that we see in a person, small motor groups of motor units alternate contracting. Again, to prevent that fatigue, but it helps to maintain some partial bit of contraction here to allow your muscles to have some sort of shape to them. So hypotonia, hypo just means it's below that normal. So decrease or loss in muscle tone. So that flaccid paralysis, flaccid flat. Muscles have a flattened appearance. Hypertonia is that increased muscle tone. Hyper means above normal. Word you're going to see over and over again. We can see it in spasticity. So there's an increase in muscle tone, which is causing those tendon reflexes to be increased. So that patellar reflex is a lot larger. And we also will see some pathological reflexes like Babinski, where you take something and scrape along the bottom of the foot. You should curl your toes, but a positive Babinski means they splay their toes out. We'll go through that again in the nervous system. Spastic paralysis usually has to do with nervous system and electrolyte imbalances. And then we have rigidity, which has an increased muscle tone where reflexes are not affected. Tetanus, like that. Clostridium tetani gets in through some sort of open cut somewhere that causes muscle stiffness and spasms in those muscles. Spasm of that diaphragm makes breathing life-threatening. The lack of breathing life-threatening, I should say. So there's different types of muscle contractions. An isotonic muscle contraction is where tension is created, but the muscle, which causes that muscle to change in length. So you go to pick something up. That muscle, after you pick something up, it's got to contract harder to move your arm back because it has extra weight. You pick up a book or something. You're moving something. We have concentric isotonic and eccentric isotonic. Concentric allows you, it creates enough resistance to move something. So that muscle is going to shorten, picking that book off that table. Eccentric is when you go to set something down. Your book uses the example of walking down a hill. Your muscles need to lengthen. And then we have isometric. The best way to think about isometric is you're holding a book out in front of you for somebody to take out of your hand. So your muscles are generating a force to be able to hold that book out, but they're not changing length. So isometric means they're not changing that length. So here's that example. Concentric, you're picking up that book. Eccentric, you're putting that book down. Isometric, you're holding it out for somebody to take. All of our muscles are made up of different types of fibers. So we have slow oxidative fibers. Slow means they contract, they're slower to contract. Oxidative, they use oxygen, so they make their energy, their ATP, through aerobic respiration. They have that slow rate of contraction. Slow oxidative fibers are very fatigue resistant. They can hold contractions for long periods of hours. So these are the muscles that are currently maintaining the posture of holding your head up, maybe keeping your back straight up. whatever position you may be in, but endurance type of activities like endurance athletes, endurance runners, swimmers, cyclists, they have these muscles trained very well. Fast oxidative glycolytic fibers. These are the largest fibers we have. Oxidative, so they can use oxygen to make energy. Glycolytic, they can also use anaerobic glycolysis. They have lots of glycogen. They contract faster than those slow oxidative. You use these for walking. You also use these for sprinting because sprinting, you can't sprint for a long period of time. Sprinting is a very quick and done activity. So they're going to allow you to quickly get something done. So I said that aerobic respiration, I just didn't highlight it. Then we have fast glycolytic fibers. Fast glycolytic, they have a lot of glycogen and they make their ATP through glycolysis. They can make that ATP pretty quickly. They break it down pretty quickly. Weightlifting, throwing. These are weightlifters tend to have larger or more fast glycolytic fibers than an endurance athlete. People who are really good at endurance activities have a lot of slow glycolytic. People who are really good at activities like weightlifting have a lot of fast glycolytic fibers. So all of our skeletal muscles are a mixture of all three types of fibers. But about half of them are going to be those slow oxidative fibers. Even though we have the other two, at least about half of them are going to be those slow oxidative. Again, your training regimen is going to determine, your genetics are going to determine. The action of the muscle is also going to determine how much of which type of fibers we have. So active postural muscles, those have a lot of slow oxidative fibers because they're holding contractions to maintain your posture. Arms and shoulders may have more fast oxidative fibers. Your legs for walking and running, they're going to have slow oxidative and fast oxidative glycolytic. So depending on where they're at in the body, it's going to help determine what we have more of in that specific area. All of our skeletal muscle fibers are the same types. So they're going to have fast oxidative, slow glycolytic, fast oxidative glycolytic. They're going to be that combination. And again, each motor unit, one motor unit is going to go to those slow oxidative. Maybe a different motor unit is going to go to that fast glycolytic. Another motor unit is going to go to that fast oxidative glycolytic. We're not going to have one motor unit that's going to have a combination of the three. Those motor units are only going to go to one type. So again, that ratio of the different types of fibers, it's genetically determined. So people with a higher percentage of those fast glycolytic are going to be really good at sprinting or weightlifting. Somebody who has more slow oxidative are going to be really good at it long distance running. We don't increase the number of muscle fibers with exercise, but characteristics can change to an extent. So endurance type exercises like running or swimming, they can transform some of those fast glycolytic into fast glycolytic, oxidative glycolytic. Endurance exercises, They can cause respiratory and cardiovascular changes that cause skeletal muscles to be better at supplying oxygen. So you can train your muscles to a point to be good at certain activities, but then again, those types of muscles that you have are going to determine what type of activities you're also good at. So there's different activities that require a lot of strength. Those are going to use fast glycolytic. The elasticity of our muscles and their connective tissue attachments are going to be very important with this whole process. So the more elasticity within our fibers, the greater the range of motion of that specific joint. Regular stretching can help to lengthen muscles, but it is a very slow process and it needs to be done daily to see any kind of change. So this is a nice chart with a picture that shows the different types of fibers, the 3 different types, the colors you tend to see them in, and their functional characteristics of these different fibers. This is a good chart from your textbook that gives us all of this information. Steroids, anabolic steroids. Anabolic steroids is a synthetic version of testosterone. People tend to use it to increase their muscle size by increasing the protein synthesis. It can be just prescribed for cancer patients, age patients, delayed puberty in males. But athletes and bodybuilders can tend to abuse these drugs sometimes because they use them as performance enhancing drugs. So they're usually taken in doses from 10 to 100 times those used to treat medical conditions. There's a lot of negative side effects to these anabolic steroids when they're taken in such high doses. Cardiovascular, it increases your blood cholesterol levels, makes your heart larger, increases your blood pressure, more blood clot formation. Forming blood clots can cause heart attacks and strokes, can cause liver cancer, can cause cysts to form. It can cause a short stature in your musculoskeletal system. Tendon injuries, acne, oily hair and skin, baldness, jaundice. People who use non-sterile needles can lead to HIV, hepatitis B and C. These people usually are aggressive, irritable, mood swings, delusions, impaired judgment. And males, because their testes usually produce testosterone. If they're injecting it, their testes aren't making it. So those testes start to atrophy, which causes their own testosterone levels to go down, which causes their sperm count to go down. They start to develop breasts and increase their risk of prostate cancer. Females, their breasts start to atrophy. So does their uterus. Menstrual irregularities, enlarged clitoris, sterility, facial hair growth, deepening in your voice, more masculine types of characteristics will start to show up. So how can we help lengthen our muscles? Stretching. We mentioned it before, but stretching on cold muscles will not increase your flexibility. You need to warm your muscles up before you stretch them. You can do it using external sources, but 10 or more minutes of muscle contractions are also a good way to raise the body temperature of your muscles that can help lengthen out your muscles. Because exercise generates heat, which will actually heat your muscles up better for more effective stretching. Some people stretch before exercise, but it's more important to warm up before exercise and stretch after. You should stretch after you exercise. Strength training. That is that process of working with heavier resistance to strengthen that musculoskeletal system. It causes stronger muscles. It increases your bone strength as well. By weight training, you are increasing your resting metabolic rate, meaning you burn more calories at rest, aka eat more food without gaining weight. Strength training prevents back injuries and other injuries. Hint, hint, keeping your core strong is the #1 way to prevent back injuries. It's not 100%, but it works extremely well. Keeping your core strong prevents those back injuries. It also reduces your stress and fatigue. If you find yourself you're always tired, strength train. It'll help. The longer you train, the longer it'll take before that lactic acid builds up. And it reduces muscle spasms. Okay, let's go on to the other types of muscle here. Cardiac muscle. We're only going to kind of mention it here. We're going to go into, you'll go into a lot more detail on it in anatomy too. So cardiac muscle is found in the middle layer of tissues that make up the heart. That cardiac muscle has its own conduction system. It is striated, so it has that actin and myosin, just like skeletal muscle. It has those Z discs that help to make that striation. They have those T tubules. And they have a sarcoplasmic reticulum, but it is a bit smaller. A main difference is cardiac muscle cells are smaller and they are connected together with those intercalated discs that are connecting one cell to the next. They have those desmosomes and gap junctions. This is what helps to allow those action potentials to travel from one muscle cell to the next. It has the endomyosum and the perimyosum, no epimyosum. A single action potential will allow a cardiac muscle cell to remain contracted 10 to 15 times longer than that skeletal muscle. And it's because of calcium from that sarcoplasmic, going back into that sarcoplasmic reticulum. And those channels that allow that inflow of calcium from that interstitial fluid stay open longer, causing that contraction to be held longer. Cardiac muscle has its own autorhythmicity cells. It contracts on its own without stimulation from the heart. Without that stimulation, it usually has a resting beat of about 75 beats per minute. It uses that aerobic respiration, so that glycolysis into the Krebs cycle through the electron transport chain to make its ATP, but I'd also mentioned it can use lactic acid to make ATP. The heart can undergo hypertrophy, meaning it can get larger. There's 2 different types. We have physiologic. This is in athletes. Like endurance athletes tend to have a larger, more efficient heart. It's larger, it can pump out more blood, which slows down their heart rate. There's also the pathologic enlarged heart, and this usually has to do with heart disease. The heart is just working harder to try to push blood through blood vessels that are no longer stretching the way that they should. Smooth muscle. Smooth muscle is that other involuntary muscle tissue. It makes up most of the organs or like walls in the organs of the chest, well, like the lungs and the abdominal pelvic area. It's also found in arteries and veins, that stomach, the intestines, the uterus, urinary bladder. It is also autorhythmic, meaning it can contract on its own. It does have gap junctions to spread action potentials. It can be stimulated by neurotransmitters, hormones, or its own autorhythmic signals. So action potentials get transmitted from one fiber to the next through these gap junctions to help spread so these contractions are done through unison. So multi-unit smooth muscle tissue. So multi-unit means we've got these nerves that are attaching to each of these individual muscle fibers here. Versus a single unit means that there's these gap junctions that are spreading these action potentials from one cell to the next. So let's go back up here for a minute. So these individual fibers have those gap junctions that allow the passage of this action potential from one cell to the next. We have these in various places around the entire body, like large arteries, airways, and the lungs, the erector pili muscles, and the hairs in the arm, muscles that connect to the iris, to your eye, to adjust that pupil diameter, the ciliary body that changes the shape of the lens to help focus light into your eye. So a single smooth muscle fiber, it's kind of tapered at the ends. You can kind of see this fusiform kind of shape that these cells make. They contain both thick and thin filaments at a ratio of 1 to 10 or 1 to 15. So 1 thick filament for 10 to 15 thin filaments. They are not arranged in sarcomeres like they are in skeletal and cardiac muscle. They contain more intermediate filaments, and there's no overlapping of these filaments, which is why it's smooth pattern. There's no T-tubules, and they have a very small amount of calcium in that sarcoplasmic reticulum. They have these little pouches or these caveoli where most of that calcium is stored. These thin filaments are going to attach to these dense bodies. Kind of look here. We've got these thick filaments and these thin filaments. You can see these intermediate filaments that are kind of helping to hold everything together in these dense bodies along that sarcolemma here. This is that they're helping to hold everything together. During a contraction, the thick and the thin filaments generate tension, and it transmits them to those intermediate filaments, which allows these smooth muscle cells to kind of contract in a corkscrew like pattern. So they kind of rotate when they contract. So they're going to kind of go in a circular type of pattern each time a smooth muscle cell contracts. And we'll see how that kind of works. So smooth muscle cells are going to contract more slowly, and their contraction is going to last a lot longer than skeletal muscle as well, just like cardiac muscle. They can be shortened, they can be stretched to a lot greater extent because of that rotation when it contracts. So as we get an increase in calcium into that sarcoplasm, that's what helps to initiate that muscle contraction, just like with smooth muscle or skeletal muscle. That calmodulin is a regulatory protein that calcium binds to. So calcium will bind to calmodulin in smooth muscle, and that'll activate the enzyme myosin light chain kinase. It's going to use ATP to add a phosphate to that myosin head. And then that myosin head can bind to the actin and a contraction can occur. It's a little bit different. Most of the time when smooth muscle contracts, it is actually going to contract because of a stress relaxation response. So say you swallow something down, it goes through your esophagus, into your stomach. It goes into your intestines. As the contents of your stomach empty into your intestines, it is going to cause that lining of your intestines to stretch. When the lining of that intestine stretches, it causes it to contract. It's one of those autorhythmic types of characteristics to smooth muscle. A stretching of those cells causes an automatic contraction to those cells. So they contract in response to that stretching. Hormones, changes in pH, oxygen, carbon dioxide levels, temperature, ion concentrations, they're all going to play a role into smooth muscle and its contractility. So hypertrophy is enlargement of existing cells. This can be skeletal muscle, cardiac muscle. It kind of goes across all kinds of different cells. Well, hyperplasia is an increase in the number of fibers. Trophy, enlargement of cells. Hyperplasia, increase in the number of cells. Those satellite cells, again, are those stem cells of muscle fibers. They can divide slowly, and they can help to repair damaged fibers. They can repair to a limited extent. It was originally thought that cardiac muscle could not be replaced. So healing that took place was exclusively fibrosis, that fibrotic tissue. But cardiac muscle can undergo hypertrophy. That tissue can get enlarged. Those satellite cells can be released from skeletal muscle into the bloodstream, go to the heart to try to help replace the tissues in there. It's kind of cool. Smooth muscle can also go through hypertrophy, so they can get larger. Think about the uterus. When a female gets pregnant, it's getting larger and larger and larger. They can also go through hyperplasia, more and more cells. Smooth muscle comes from pericytes, which is the stem cell of smooth muscle. And smooth muscle has the greatest ability to regenerate. I think I wrote this once before, but it's smooth. Skeletal, then cardiac. This is the order. Smooth muscle has the largest ability to regenerate, then skeletal muscle, then cardiac muscle has the least. So here's the different types of muscles, the 3 different types of cells, their different features, their appearance, where they're found, the connective tissue it wraps around them, their typical length, whether they have the contractile proteins in a sarcomere, have sarcoplasmic reticulum, the T-tubules, junctions, autorhythmicity, where they get their calcium from, regulatory factors, how they contract, regulation, capacity for regeneration. So all of our muscles come from the mesoderm during embryologic development. That mesoderm is going to go through segmentation that is going to form somites. We see those first somites at the 20th day of development. Eventually, we're going to have 42 to 44 pairs of somites by the 5th week. Those somites are going to create a myotome, which is the skeletal muscle of the trunk and limbs, dermatomal mesenchyme, which is that connective tissue, and then a sclerotome, which is going to give rise to the vertebrae and the ribs. Cardiac muscle also comes from that mesoderm. Smooth muscle also comes from that mesoderm. So all muscle comes from that mesoderm. You can see all these little red circles. Those are giving us those different somites that are developing, which are eventually going to give rise to that dermatomal mesenchyme, that myotome, and that sclerotome. So between the ages of 30 and 50, humans slowly start to lose their muscle mass, and it gets replaced with connective tissue and fat cells, those adipose tissue. We lose about between 30 and 50, we tend to lose about 50% or 10% of our muscle mass. Could be because of a decline in physical activity. And it causes a decrease in strength. Your muscle cells, also that reflex is going to slow down. You're going to lose some flexibility. You gain more slow oxidative fibers. You lose another 40% between the ages of 50 and 80. So by 80, you've lost about 50% of your muscle strength. Most people don't see that they've lost muscle strength until they're between 60 and 65 years old. Your lower limbs, your legs weaken before your arms do. Again, exercise is effective at any age. That aerobic and strength training is effective and can slow down or even reverse age-related muscle decline in strength, that muscle performance. So exercise is good for your muscles to retain your muscle strength. So let's look at, there's some neuromuscular diseases. Neuromuscular means it's between that neuron and that motor and that muscle fiber. And then we have myopathies, which is problems with diseases with skeletal muscle. So myasthenia gravis, I mentioned that we would talk about it when I pointed it out earlier in the PowerPoint. It's autoimmune, and it is where your immune system is coming in and attacking that neuromuscular junction. So it is blocking those acetylcholine receptors. Eventually, you're going to lose those acetylcholine receptors because that immune system blocks is breaking it down. Most people with myasthenia gravis have tumors in the thymus, one of your lymphatic organs that helps with your immune system. So as it progresses, muscles become weaker. They fatigue a lot quicker, and some of your muscles don't work. It's more common in women. It usually onsets between the ages of 20 and 40, and if men get it, it sets in between 50 and 60. Your muscles of your face and your neck are affected first. So double vision, difficulty swallowing, difficulty chewing and talking will come next. They use those anticholinesterase drugs to try to keep that acetylcholine more readily available at that neuromuscular junction. They'll also use steroids drugs like prednisone to try to reduce those antibody levels in the blood to prevent that attacking at that neuromuscular junction. Muscular dystrophy. It's a group of disorders. There's many different types. And again, they're inherited, and it's where your muscles are being destroyed, and it's a progressive degeneration. Duchenne's muscular dystrophy, it's the most common, and it's the saddest type. It's caused by a mutation on the X gene, which means it affects almost exclusively males, since they have one X chromosome. 1 in 3,000 or 3,500 babies, male babies, are usually born with Duchenne's muscular dystrophy. It starts to develop between the ages of 2 and 5 when Parents start to know that their children starts tripping more often, they're falling, they used to be able to run, now they can't run anymore, jump, hop, play. By the age of 12, they're not able to walk. And by the age of 20, they usually die because of cardiac or respiratory failure. So that genetic code for dystrophin, which is one of the proteins found at that sarcomere, gets mutated. So there's no dystrophin there at that sarcolemma, which causes those muscles to tear upon contraction. They can treat it with steroids to help strengthen those muscles. There's some other drugs that can go for that targets more that gene mutation, creatine supplementation to help improve strength, but it's still pretty sad, especially since it affects kids. A muscle spasm is an involuntary contraction of a single muscle in a large group of muscles. A cramp is a painful spasmodic contraction. Not enough blood flow, overuse, low levels of electrolytes such as potassium can cause muscle cramps. If you've ever had like a Charlie horse in your leg, that's a cramp. A tic is a spasmodic twitching that is an involuntary twitching by muscles that are normally under voluntary control. A tremor is a rhythmic, involuntary, purposeless contraction that kind of provides a shaking movement. You usually see tremors in people's hands where they have this slight shake going on in their hands when they're trying to do something. That's an intention tremor. Resting tremor is when they're just sitting at rest and their hands are shaking. A fasciculation is an involuntary brief twitch of an entire motor unit that is visible under the skin. It's not associated with movement of any affected like muscle. It can be seen in people with multiple sclerosis or amyotrophic lateral sclerosis. And fibrillation is a spontaneous contraction of that single muscle fiber that's not visible, but it can be recorded. Could be causing the destruction of motor neurons that are going to that muscle group. Exercise-induced muscle damage. So after you do some intense exercise, it can cause small tears to that sarcolemma, damage to those myofibrils, disrupt some of those Z discs, and it increase blood flow to the area, more myoglobin, enzyme creatine kinase. These are things that you can kind of monitor through blood. Then there's DOMS, delayed onset muscle soreness. Stiffness, tension, swelling. If you've ever exercised, done some intense exercises you don't normally do, go to bed, the next morning you wake up and it's hard to move. That's a delayed onset muscle soreness. And there's not a complete understanding of what causes DOMS, but it can onset 12 to 48 hours. It's usually from 12 to 48 hours. So you first notice it maybe 12 hours after you do something, and it can last up to 2 days. Muscles do undergo repair. New regions of that sarcolemma can be formed to replace those torn sarcolemmas, and more muscle proteins can be made to help fix any of those damages. And we have made it to the end of muscle tissue in chapter 10. Thank you very much for listening.